here this evening. The story I want to tell you today essentially spans about 100 years, and the work I want to share spans probably in the last 20 years. And it's not just my work. I've worked with a lot of colleagues and friends on this, so it's a collaborative effort, so I have to give kudos to them as well for this work. So essentially, as many of you may know, Corymbia calophylla, or Marri, is a keystone species here in the southwest. This gray area here shows the range of that species, and is considered a keystone species mainly because it is obviously a food source and nesting location for many black cockatoos, some of which are endangered, as well as other birds, and obviously the beekeeping industry as well. So honey is precious. A related species has this small range down here near Albany and plays a key role in this story, and that is Corymbia fusifolia, or the red flowering gum, quite similar but with red flowers. And the story essentially starts with this species back in the 1920s, when stem cankers were first observed in Kings Park. Now, this is a view from Kings Park around the 1920s, maybe 1930s, and essentially Corymbia fusifolia was planted along the main avenues at that time. I forgot what the street names are, but during this time, these huge cankers started forming and completely demolished those trees. So they removed them and planted them with Corymbia citriodora, which is actually an East Coast species, and that's what you probably see today. I'm sure some of you probably already know this. But they took a sample from the trees that were essentially affected by this disease and isolated a fungus called Sporotrichum destructor, and that was unofficially described at this time. Because this fungus was only observed in these amenity plantings in Kings Park, no one really worried much about it. They got rid of it, replanted, and everyone moved on. About 20 years later, these, similar looking stem cankers were found in Marri in Pickering Brook. And they took samples again and found it to be the same fungus from previously, the 20 years previously. Again, this was popping up very sporadically. No one really cared about it, and everyone moved on. Fast forward to the 1990s, or the early 2000s, so almost, 50 years later, everyone started noticing these around pretty much in various areas around the southwest. So this is what we would call a stem canker. This is quite an aggressive, advanced one. Essentially what happens, this fungus enters the bark starts splitting, and that it just starts ripping apart. I essentially compare it to almost being, strangled in a sense, because it wrings the stem or the branch where it's infected and kills off whatever's above it. You can see one on the right here, which is starting to form. And these are popping up everywhere in all parts of the tree, so higher up, as well as in branches. And many of you know that when you wound Marri, you get the chino, or the sap coming out, which stains the bark as well. So at the time, a colleague of mine, Dr. Trudy Papp, who I think some of you may know, started this work and wanted to know, in the early 1990s or 2000s, was this fungus the same as what was observed in the 1920s? So she took samples and essentially officially described it as Quambularia coriaca, which causes these cankers, as you can see here. Here's a photo down here where essentially that fungus is sporulating and coming out of the plant, and what it looks like if you try to culture it. What's unique about this pathogen is it hasn't been found anywhere else in Australia, and so we believe it's actually endemic to this region. And in most cases, endemic pathogens don't cause such destruction or mortality. I mean, you think of Phytophthora cinnamomai or dieback, that's an introduced pathogen and has caused declines in a lot of different species in the Southwest. So the question arose, well, if this is an endemic pathogen, why is it causing such a big problem? So again, Trudy wanted to find that out and wanted to know what is the actual impact of this canker disease, as you can see here in this picture. So she set up 3 sites and did a long-term survey from 2001 to 2014. And each site is in a different location, with different climate, with different soil, with different vegetation, because she wanted to see, does any of that influence the disease? And she monitored these sites, again, for that period of time. And at each site, she had essentially one, which was like a forest block, like a healthy area, and an area that was disturbed, or what we say anthropogenically disturbed, essentially pressure from humans, whether that's fragmented, it's a paddock, it's a road edge, that type of thing. And one of the main findings, this is just a photo from, this is the same tree over 14 years. So at the original time, it was quite a large canker, but by the end, that tree had succumbed to that disease and died, unfortunately. And this was seen in different individuals as well. But one of the main findings was that habitat fragmentation is a driver of this disease. So, for example, this picture on the left shows what we would say it's fragmented. There's a road in between the forest. So these are forest edges in comparison to what you would walk into a normal natural forest. And this figure here in the middle just shows you the percent of trees in total, up to 100%, and those along the disturbed edge and the forest edge. And along the disturbed edge, about 65% were healthy, 35% were not healthy, they had cankers. By the end of 2014, there were more cankers, less healthy trees, and this dark bit here is about 10% had succumbed to those cankers and died. In the forest area, we had about 90% healthy trees. There's still some cankers, but not as many, and that percentage stays the same as by the end of 2014. But we still have some death, but then this dark blue area is some death by other causes, whether that's drought or anything like that. So going on from this, I wanted to show, this is an older photo, so it's not really up to date, but this just shows Australia and the modification of the landscape. So obviously the dark green is wilderness, nothing's really been disrupted or modified. You can see urban centers are dark red. They're highly modified areas, so quite urbanized. But if we zoom in to the southwest, you can see that most of it is quite modified, as I'm sure everyone, when you drive through, realizes. We're talking about little forest patches that we think are important, and they are. And essentially this modification overlaps the range of Marri. So it's being high, there's a lot of pressure on Marri from habitat modification or fragmentation. So knowing this, Trudy's next question was, how widespread is it? Is it just in those pockets that she surveyed, or is it all over? So again, Trudy's been the baseline of a lot of this work. She set out and sampled 62 sites across the range. For the time being, I just want to focus on the circles here. So these are all the sites she went to, and she measured the presence or absence of canker. The white circles are sites where there was no canker. So of the, I believe it was, I say at 62, about 8 did not have canker, and those were found in the northern part, as well as down here, kind of on the eastern, I guess, end range of Marri. The color-coded areas are yellow. Less than 20% of trees had canker, and as they get darker, the more that that they had. So we wanted to know what other factors were driving disease. And essentially, she went around these sites, also looked at climate, so rainfall and temperature, but also interaction with other pathogens such as Phytophthora or dieback. And so we're going to go through that just step by step here. This figure shows the proportion of cankered trees against average rainfall over a 30-year period. And essentially this tells us that as the rainfall increases, we get an increase in cankers. With temperature, we see the opposite trend. As temperature increases, we see a decrease in temperature. And if you think about fungi or mushrooms, you tend to only find them in wetter, cooler areas and not in the middle of summer, for example. So this climate is a perfect, I guess, breeding ground for the fungus. At each of those sites, in collaboration with a PhD student at the time, Louise Cruiser, they surveyed the soil and looked at what Phytophthora species were present. And out of 62 of the sites, 34 had some Phytophthora species. Many of you are probably familiar with Cinnamomi, but there are other species as well that can cause disease, and some that aren't that bad. But essentially this photo here is a root mass of Marri that's healthy, and this is what a root mass looks like infected with Phytophthora cinnamomi and another species, Multivora. So you can see it completely destroys the root mass, and that causes issues for nutrient uptake and other things like that. When they looked at the, again, proportion of cankered trees, where Phytophthora was absent and where it was present, you see a higher proportion of canker where it's present than where it's absent. So essentially, Phytophthora and this canker disease are interacting and essentially making it worse for Marri. So she put all of this together and essentially modeled and tried to predict where we could find canker based on climate and other diseases. So these colors here show the probability of canker, and the white areas essentially show where it's less likely to occur, and it's more likely to occur down south. And if you've ever driven down south and you just have a look outside on the road edges, once you see it, you can't unsee it. It's actually quite sad. And I did a lot of this work almost 10 years ago, and I've driven down recently, and I think it's actually gotten worse, unfortunately. Okay, so leading on from this, again, Trudy asked the question, what is different about these disturbed areas? And she wanted to inspect those at a finer scale. And so what she did, instead of doing this at 62 sites, she minimized it, looked at 17, and essentially set up a gradient of different transects. So this first transect here is what we would call a highly disturbed edge. It's bordering some type of farmland or grazing land, essentially some sort of land use, and on the other side is a road. So it's being impacted by two types of disturbances. On the opposite side of the road, you would have the forest edge, so that's experiencing one type of disturbance. And then she wanted to know how far away from that road does it improve, and so she looked at 50 meters from the edge and then 200 meters from the edge. And essentially, what we found, again, this is the proportion of cankers, and at the disturbed edge, almost 50% of trees had canker, but when you went to the forest edge, that dropped to almost half, about 25% or just below. And then as you walked into the forest, it got lower and lower. So essentially, it's showing that double exposure to disturbance is causing some sort of pressure. If we look at the number of dead trees along the same transects, there are more dead trees on those disturbed edge than even when you go into, or the forest edge, and then there's not very many dead trees as you go into the forest. So again, this proves the fact that habitat fragmentation or disturbance is driving canker incidence in this area. But again, what is different about these disturbed areas? So one thing she looked at is stand structure. So before we get into her results, this is essentially what that disturbed edge looks like. You have some sort of agricultural land and the road edge, and that's what it looks like essentially in comparison to what a forest looks like. Just by looking at the photos, you can see that there is some sort of difference. So she measured the total, or the stems, number of stems per hectare of all species, and essentially the density, or the number of stems per hectare along that disturbed edge, was much higher than any of the other transects. And when you looked at all of the Marri of those total amount, essentially the percentage of Marri was much higher. So what we're seeing is a much higher density of Marri on these disturbed edges than in comparison to a forest. And this is essentially seen with, I guess, basal area, so essentially how much area a tree is taking up. That is much higher with all species on this edge, but again, Marri are taking up most of that space. So just to summarize this bit, essentially what we're seeing with an increase in disturbance, there's an increase in canker disease. An increase in disturbance, we're actually seeing a greater density of Marri, which sometimes doesn't make sense, but an increase in Marri density, you see an increase in canker incidence. I guess you could compare it to, I don't want to bring it up, but COVID times. You don't want to be close to people who are infected, you want to be further away, and so in the forest, you're nice and spread out, you're probably not catching disease from others, but when you're close to each other, it's much easier to catch something. But what's interesting with this is Marri is considered a quite tolerant species to drought, to other diseases. So it's kind of been selected to grow on these disturbed areas, but it's become so bad that they can't survive anymore, essentially. So most of this work that Trudy did was above ground, and my work came into, I wanted to know what was happening below ground. I mean, we all know plant health is related to soil health and vice versa, so it's quite an intricate relationship. And what affects the plant above ground can affect the plants below ground. And there's a group of fungi, which I'm sure you're all familiar with, that I study essentially, and those are mycorrhizal fungi. I show this figure here with the pine tree because it just shows the relationship quite nicely. But you have mycorrhizal fungi like Amanita muscaria, which essentially attach to root tips and help the uptake of nutrients and water for the plant. Globally, 90% of plants actually associate with these fungi. So you do have some that form fruiting bodies, but the majority of them just chill out in the soil, and they form this almost, I guess, protective cap over the roots and grow out into the soil. So they increase the surface area and allow the plant to uptake more nutrients, and the plant gives them food in exchange. So my question was, how does disturbance change these really important mycorrhizal fungi communities? And so I went to the same sites that Trudy did, and this time looked at a disturbance gradient as well, but I didn't look at the 50 meters because there wasn't much difference. So I went to all of these sites, took some soil samples, and unfortunately, unlike trees, you can't just go out and measure them. You have to do all this molecular work to extract the DNA of fungi, and I can't show you a nice photo of changes in fungal communities, so I have to show you this figure, which is essentially the presence and absence of different fungal species. And in this case, each point is a soil sample, and it tries to compare the community from this sample to the community from a different sample. So the closer the points are together, more similar they are, and the further apart they are, the less similar or more different they are. So to take from this figure is essentially the yellow circles are all the samples from the disturbed edge, and the green are from the forest, and those don't overlap at all. So that essentially means that the communities are completely different, and so something is happening below ground. At the same time, I also measured soil nutrients, and what we see along that disturbed edge is we have a much higher concentration of those different nutrients. So those were the main ones, but I measured a whole suite. And essentially you can think of along those disturbed edges, if you're on farmland, you're probably getting runoff from nutrients, fertilizers, whatever. And even on the forest edge, you're probably getting some sort of effect from the road as well. So I also tried to put this together and essentially model what are the drivers below ground causing canker incidence. And before I get into that, this is just showing the number of species of those mycorrhizal fungi across that disturbance gradient, and essentially we see much lower number along that disturbed edge in comparison to in the forest. So again, we're losing diversity, we're losing species. Some of these species could be important for Marri to help them uptake nutrients and even protect them from pathogens as well. So I put all this together and I measured some other aspects about the disturbed edge. I don't know if that's quite clear, but along these disturbed edges you see a lot more leaf litter in comparison to within a forest. And when I put everything together, the proportion of trees with canker increased with leaf litter depth and then decreased with the number of mycorrhizal species. So the more mycorrhizal species you have, the less likely you are to get canker. So this seems kind of odd. So what I want to do next, before I finish, is take everything that Trudy and I and some other PhD students at the time did and tell you what I think is happening.
Whoa, very sorry.
So you go into a forest, there's probably Marri, Jarrah, Banksia, a whole bunch of other species. Everyone's happy, you have your black cockatoos, it's great. And then we decide to build a road through the forest, and so now we get that forest edge along a road. And what happens is, in most roads you get this, like these ditches on the side, and you probably get water runoff into those ditches. And even that water runoff changes the properties of the soil. Mycorrhizal fungi are highly sensitive to those changes. Plants are sensitive to those changes. And so Jarrah, which is quite sensitive, or much more sensitive than Marri, starts to disappear over time. And Marri goes, oh, I can handle this, and continues growing. But they get a little bit stressed and start forming a few cankers. Some of these cankers might cause death of some branches. Those branches fall to the ground and you start getting this layer of leaf litter. Now we decide to clear the land and, I don't know, put a vineyard there. So now all we have is this little stand of Marri left over, being pressured by the road, being pressured by the vineyard, and Marri are still able to survive, and over time there's all the other species disappear, so you get this high density of Marri appearing, but now these cankers are forming. They can't fight against this anymore. And more cankers appear, more tree death, more branches fall, and now you get this huge layer of leaf litter. And if you've ever dug through leaf litter and into the soil, that soil is quite moist, it's quite different from any other soil, and so just having that layer is completely changing the soil chemistry and properties of that area. And so all these extra stresses are just causing the tree not to be able to cope anymore. It's like if you're stressed and then you get a cold, you never fight off your cold because you're stressed, and it's this terrible feedback onto itself. So I'm going to end with hopefully a positive note. It's been a lot of doom and gloom. And what can we do? Well, during the time we were doing this work, we ended up collecting seed from all over the range, from 18 provenances covering 170 families, and essentially grew 12,000 seedlings. Doesn't look like 12,000 in the photo, but there's a whole bunch of trays. And we planted 4,000 seedlings at 3 different locations. And this trial is still ongoing. We planted in, must have been 2015, 2016. And this is probably what it looked like a few years ago. They're probably much bigger now. But the whole point of this trial is to assess variation in morphology and physiology between the different provenances, so different local, I guess, genetics, essentially. How they perform under different site conditions. So for example, if you collect plants that live in the northern part of the range, it's much drier, warmer, how do they survive in a cooler climate? And vice versa, if you put ones that are used to a cooler climate up into a hotter climate, how do they survive? And this would be important for things as we start getting higher temperatures and less rainfall. There is also looking at the variation in susceptibility to this canker fungus. So there's a potential that there will be individuals who are tolerant or resistant to the disease. And with those individuals, we could potentially develop disease-resistant limes that we could replant in these areas that are highly pressured by that disease. So this is ongoing, and we just need to monitor it and see what comes from it. So thank you so much for listening. This, obviously many of you may know Trudy, but I had my advisors, Trina Burgess and Giles Hardy, and the other PhD student, Louise Cruiser, as well. So thank you so much.